previously about the innate immunity, so the cytosis complement system.
She was somewhat like the
Started
brain.
talking about the acquired
She
or
was
adaptive
somewhat like
type
the
of
brain.
immunity, one that's storing maybe the lymphocytes. Told you that there are two main types of lymphocytes, B and T, respectively from the bone marrow and to the thymus gland. And they are responsible, as shown there, for they respectively B cells for humoral immunity. That's what we talked about last time, the production of antibodies, and the role that antibodies play, specifically binding to certain antigens. And then cell-mediated immunity is what we're going to start talking about now. So we finished by just summarizing the main features of humoral immunity, major defense against bacteria, and so on, which we talked about last time. Now let's pick up here with cellular immunity, or cell-mediated immunity, same thing. Major defense against viruses, against cells which have undergone abnormal changes, cancerous cells, or cells which are normal in themselves but have come from somebody else. And so to the body concerned, they are foreign cells, transplants. Major cells are the T cells. B cells are of different types. We talked last time about B cells, which can mature into plasma cells. We talked about memory cells, which are, I call them half switched on. They're able to respond better to secondary exposure. In the same sort of way, T cells have also got many subtypes. There are main ones are cytotoxic T cells, and I'll explain what all this means in a moment. There are helper T cells, and subtypes of these subtypes. I'm not going to talk about these in much detail. And also memory cells. So you're able to retain a memory through memory B cells and also through memory T cells. Both B cells and T cells have to do the 3 R's. So we saw that B cells recognize because they've got antibodies on their surface. They respond by differentiating, producing antibodies, and then they remember. T cells have to do the same 3 things, although they do it, as we'll see, in a somewhat different way. And one of the main differences between the B and the T cells is that T cells require something called antigen presentation. When a B cell encounters an antigen, from that diagram I gave you last time and from the description I gave you, you've got B cells floating around, antigens also floating around outside the B cell, and by chance every so often they're going to come in close enough contact with a B cell with the right specific antibody binding sites, and they can bind, and the rest of the story as we talked about previously. T cells, you can think of them in a way as being, I don't know, fussier, more not selective isn't the right word, but they need an extra bit of help, if you like, in order to recognize their particular antigen. And what is involved then in antigen presentation? And this is summarized in figure number 40. Here's the T cell that we're going to be talking about, and here is an antigen presenting cell. Now, not all cells can do this. There are what are called professional antigen presenting cells. And these, or at least 2/3 of these, we've already come across. B cells, macrophages. We've seen other functions of these cells, but in addition to what we've already talked about, they can also act as presenters of antigens to T cells. So here's yet another example of interaction between the different branches of the immune system. The one we haven't talked about yet, but very important, and maybe the most important of these, are things called dendritic cells. Have you ever heard of these? Recently? Yes? Well, you're taking immunology. Anybody else heard about these recently? So they're very in the news nowadays. If you want to know more about them, just put in some of these names and and you'll get a lot more, both at the sort of general level and also in more detail. So I just mentioned briefly, I didn't mention these people's names previously, but the first two, Briefler and Hoffman, they were involved, they were rewarded for making these discoveries about activation of the innate immunity, this idea of how cells are able to recognize through these receptors and so on their surface certain characteristics of things like bacterial walls, the toll-like receptors and so on that I didn't go into. And then the relevant one here is Steinman, who discovered the dendritic cell and its role in adaptive immunity. I don't know if you heard the whole story, but very sadly Steinman died 3 days, I think it was, before the announcement that he had been awarded the Nobel Prize. This was just a dramatic, almost a bit of a and the rules of the Nobel Foundation say that Nobel Prizes cannot be awarded posthumously after someone's died. You can't think, all the work they did was great, now it's given the Nobel Prize. You can't do that. So strictly, he had died just 2 or 3 days beforehand. But they did let it go through because they didn't know about it at the time. So it's a whole sort of human interest story there as well. So what happens then? These cells, dendritic cells, etc., antigen-presenting cells, they take up the antigen into them, and then they partially break it down into fragments. And these fragments are then presented on the surface of this presented cell, together with a molecule called an MHC protein. MHC stands at the bottom there for major histocompatibility complex. I'll say a little bit more about those in a moment, but just for now, I think it's a presenting protein. And it's this combination of the MHC protein together with the antigen fragment that is recognized by the T cell. And the T cell has receptors on its surface which are able to recognize the combination. So the B cells have receptors on their surface, which are antibody molecules, and they're able to recognize the antigen that goes part. T cells have got receptors on their surface. They're not antibodies, they're proteins or T cell receptors. And these, again, you have an enormous number of possibilities of recognition. Remember that number, which I didn't really know for B cells, but I said it's something in the order of tens, maybe hundreds of millions of different antigens that the B cells are able to recognize. We can make tens of millions of different antibody molecules. We also do make probably around about the same number or maybe even more of different T cell receptors. So again, very highly specific to anything that we might encounter. That's not the whole story, it's even more complicated than that. In order for recognition and response of the T cell to occur, you not only need the presentation of the antigen fragment, a peptide, which has been broken down from the protein antigen, and the MHC. You also need a whole group of what are called co-stimulatory molecules, which are present both on the presented cell and on the receiving cell, the T cell. And these have to be complementary. And I want to just mention those because they're very important, but I'm not going to talk more about those. There's different numbers and letters and things which you can look up if you need to. And the consequence of this presentation is that this T cell will then increase in numbers. It will become more active in certain ways depending on which type of T cell it is, and then it brings about a response. I don't know if it's helpful to you to have some of these little analogies that I give you. This is a rather silly looking one, but it helps you to understand the matter. We go something like this. And this is all color-coded to fit in with this. So let me explain what this is all about. You can go to There's certain parties that one goes to, I think I'm sure one does. When you go to somebody's house, you go into the kitchen, you open the fridge, you grab a bottle of beer and you drink it straight out of the bottle. But there are other sort of parties that you might be invited to where this sort of behaviour would be absolutely not the thing to do. And I'm looking at those ones. So you go and not only do you not grab the bottle for yourself, somebody has to pass it to you. And not only do they not pass you the whole bottle, they pour it out into nice little units for you to take one of those. So you've got here, as you can see, the colour coding over there. The antigen, or what is being presented, is the wine in the bottle. It's broken down into smaller parts, and then it is presented by this fellow here, is the presenting cell, and he has a tray, the MHC molecule, on which is what he's actually giving you, the small portion of the wine, and then you have my hands. You know, if you're dressed properly, you've got the right gloves on or whatever, and only then are you able to complete this transaction. So it's a little bit sort of odd, but you know, this is basically what's going on. Yeah. So what are these MHCs? The silver tray over there, what is it? Very brief summary of some of the things about these MHC molecules. MHC, or MHC genes, stands for this. They code for these proteins. And these proteins are present in most cells in the body, have them on the surface. And how many of them do we have? Again, this number, I'm not sure how accurate this is, but it's a very big number, very, very big number of different MHC proteins in the population as a whole. For any single individual, it's a relatively small number. Can you update me on these numbers, or do you still know? Sorry to put you on the spot, but still. Okay, so we're not sure about the but it's big, the first number. Yeah, and over here we've got a couple of people. Would you, sorry again? No, it's about, anyway, that's just a big number. It's a very big number in the population as a whole, and a small number for any one individual. And one of the consequences of having all this enormously large number of different MHC molecules in the population as a whole is that these molecules are acting as identity markers. So this is part of the reason why when you're doing transplants, you have to try and get as good a match as possible. Because if everything was different in the recipient and the donor, or if all their MHCs were different, then, it wouldn't work, probably. If they were all the same, it would be great. But probably, you're going to have to go somewhere in between to get as good a match as possible. I'm not really going to talk about that now. I'm just talking about that other role, which is as presentation molecules. There are two main types of these MHC molecules, class 1 and class 2. And that's really all I'm going to say about them. The MHC1 is present on nearly all cells in the body, whereas the MHC2 are present, in addition to MHC1, on these professional antigen-presenting cells. So what I have done in the next figure, which looks well complicated, but this is, I hope, going to help you to put together these different branches of the immune system. So the simple map goes something like this, but to add now a bit more to this, that's what we're looking at here. Let's start with what I think is maybe in a way the simpler bit to explain, or to remind you of, the humoral immunity branch on the left-hand side here. So antigens recognized by antibodies on the surface of B lymphocytes. These B lymphocytes are then stimulated to multiply, most of them turn into plasma cells, which produce lots of antibodies. Those antibodies then go off and specifically bind to their own particular antigen, they label it, and then they can act as opsonins, they can activate the complement system and so on, we talked about previously. You're left with, after all, this is after the problem's been cleared away, you're still left with some memory cells which remember that one particular antigen, and then you see it again, they work more quickly and more effectively. Okay, the rest of the figure over here is talking about the role of T cells. And I said there are two main types of T cells, helper T cells and cytotoxic. So first of all, the cytotoxic ones here. These are able to respond to cells which have become altered in some way. So the cell has become cancerous, if it's altered in the sense to you, it's foreign to you, it's coming from somebody else, or if the cell is infected by a virus. When a virus gets into the cell, it tries to take over that cell's machinery to its own ends, and it's sort of in a way no longer your cell, it's been taken over. So just about all cells have got MHC1 on their surface, and this then, the thing that's causing the problem, the foreign protein or whatever, broken down. This should actually be an antigen fragment here, or these two here. That's not the whole antigen. And these are presented together with those co-stimulatory molecules, which are not shown here, to a cytotoxic T cell. And this cell responds by producing certain molecules. I'm just giving you an example of one molecule here called perforin. And the name suggests perforation, punching holes and so on. And that indeed is what this molecule does. This molecule, which comes from these activated cytotoxic T cells, punches holes in the altered cell. And various enzymes can move through these holes in the membrane, and the end result is you get stimulation of apoptosis in the altered cell. You're familiar with apoptosis? Programmed cell death. And that's what we mean by cell-mediated or cellular immunity. In the center here, helper T cells, I put them in the center because they have a very central role in all of immunity. Helper T cells, again, they've got their own specific T cell receptors, and these respond to antigens which are presented by the antigen-presenting cells, the professionals, the dendritic cells, etc., together with an MHC2. So remember, there's two different main classes of MHC1, MHC1 on nearly all cells in the body, MHC2 and 1, although it's not shown here, on 2 is only on the professionals. And these helper cells respond by producing whole families of different cytokines. And there's different subtypes of helper T cells that produce different subfamilies of cytokines we can learn about. I think the main point here is that these cytokines are essential for normal overall immune responses because, follow the dotted lines here, some of these cytokines feed back on helper T cells and make them more effective. Other cytokines feed onto the B cell part of the story. They enable B cells to do their recognition, responding, and so on better, more effectively. Some of the cytokines work together with cells which are typically thought of as being part of the innate system. They help with phagocytosis, they produce inflammation. So another example of interaction between the innate and the acquired system because of these cytokines here. And then another group of cytokines help with the activity of the cytotoxic T cells and their role in causing cell death. So you can see, I hope from this, that the health of T cells are really central to the whole immune system. And this is one of the main problems that is seen in people with HIV, which seems to focusing on health of T cells. If you wipe out the health of T cells, you are essentially affecting all of the immune system. And there's also memory cells, helper T memory cells, cytotoxic memory cells, as well as the B cell memory cells. And I just, in the last couple of figures in this block, I just want to give you a couple of practical applications. And this first one here is to do with these memory cells, immunological memory. It's really putting in a graph with what I've been saying. If you measure the size of somebody's immune response when they're exposed to a particular antigen X, you could measure it, for example, in terms of antibody levels in the body. You can see that when they're exposed to X at time 0 here, it takes maybe 2 or 3 weeks for antibody levels to reach a peak. That peak is a small one. So it's a slow and small response. So if you had your flu shot, it probably tell you it's not going to protect you immediately you walk out of here. It's going to take a while, a couple of weeks at least. But if you're exposed to the same antigen second time, the timing here not significant, sometime later, weeks or months later, then because you have these memory cells, the response, as you can see here, is much quicker, within a few days rather than weeks. It's obviously a bigger response, and it will also last for longer. So this is the principle behind the memory cells, or why they're significant. And then the last one here, or second one rather, is the practical application of immunization. So it ties in with what we just talked about. It goes into a little bit more detail and talks about active and passive immunity. So let me just explain what's going on here. What I've been emphasizing most of the time is that if you are exposed to a particular antigen through some disease process, then you can respond by producing antibodies. So this is called active immunity. If you're exposed to a disease, you yourself produce the antibodies because your B cells are activated and so on. It's slow to begin with, but the second time around it's relatively quick, and it lasts for a relatively long time. And the reason why this lasts for a long time is because of these memory cells. But if you want a quick response, can you do this artificially? Well, yes, of course, this is what we've just been talking about. Vaccination, immunization. And when you go and have the flu shot, you're being exposed to the antigen, but rather than having to suffer through the bout of the flu first in order to produce the antibodies, the antigens have been treated, they've been heat treated or whatever, so that they are no longer active in giving you the disease, but they still are antigenic. So you produce the antibodies. So this is by an altered antigen. It's no longer disease producing, and they're often reassured by the people giving these injections. They say, a lot of people think you're going to catch the flu by having the flu shot. And they say that's not true. Because it has been all been organism's been altered in such a way that it will cause you to produce the antibodies, but not to give you the disease. And so the purpose of this is to prevent you getting the actual full-blown disease at a future exposure time. Passive immunity, on the other hand, is when a person acquires antibodies which have been made somewhere else. In active immunity, you yourself are actively producing the antibodies. In passive immunity, you could have the antibodies which have been made in somewhere else, in an animal or in a person, could be injected into you. This can occur naturally in babies, and some antibodies, not all, but some antibodies can move across the placenta, so even before the baby is born, or it can acquire immunity from the mother's milk. One of the reasons why breastfeeding has advantages for the baby. So these antibodies have come from somewhere else, and they're going to work immediately. You don't have to go through this long process of switching on your B cells and so on. You have been given the end product, so they'll work straight away. But it doesn't last for very long. Why not? Because in active immunity, when you switch on your own ability to produce antibodies, you can keep on producing more and more. When you're given the antibodies, these are protein molecules. Proteins get broken down over a period of days, weeks, and so on, are in the body. So once they're broken down, unless you have a second injection, it's gone. The immunity's gone. But it is advantageous because if you've already got some infection, you don't have time to wait for the weeks that it might take for you to produce the antibodies. You could give somebody the antibodies straight on to help protect. I'm not going to put up figure 45, that's the glossary, which may help you with some terms that you're not familiar with. Now I didn't show them the details. So, just to summarize then what we have talked about and will be talking about, figure 46, we talked a little bit, not much about transport of oxygen, the main role of red cells, talked a bit more about the immune role for white blood cells. And the next one, to make a start on today and carry on next time, is the role of platelets. And the major role of platelets, as it says here, is in hemostasis. So that raises a few questions. What is hemostasis? Prevention of blood loss. It asks you what is homeostasis. Any answers to that? Or do we need to just throw out a definition of homeostasis?
Just the body trying to stay within, an optimal range.
Okay, the body's way of staying within an optimal range for things like temperature, pH, etc. So I've said this before, but again, don't get mixed up with these two similar sounding words. Why is hemostasis necessary? To prevent blood loss. Blood is a fluid, and it needs to flow most of the time. But if there's damage to the blood vessel wall, okay, the blood's going to keep flowing unless you do something about it. And you'll also lose blood. So those first two pass over fairly quickly. What I'm going to be spending more time on is how is this achieved, and what has this got to do with platelets. So in the same way that when we talked about immunity, is largely white cells that we were talking about, but inevitably one needed to talk about some other things as well. The same with this one. We're talking about the role of platelets, but we're also going to be talking about other factors which are crucial to prevent blood loss. So just a bit more background first of all. It might be helpful as we go through this to think in your mind of a balance. And on one side of this balance, you've got factors which keep the blood fluid. And this is what you want most of the time. You've got to keep the blood fluid to keep it flowing. And anything which favors this is called anti-hemostatic or anticoagulant factors. But if some damage does happen, often it will be fairly localized. And what you need then is various things to spring into action in that local damaged area, not everywhere in the body. That would defeat the whole purpose. What you want is local prevention of blood loss where it's happening. And these are pro-hemostatic or pro-coagulant factors. So what we're going to be talking about in part is changes in balance between things on the right-hand side and the left-hand side of that figure. How can we prevent blood loss? Well, blood flowing through blood vessels. How about changing the state of the blood vessel? And that's the first one on this list here. And we're asking how, in general, can we prevent blood loss? Vasoconstriction. Remember what the blood vessel looks like. It's got smooth, well, not all blood vessels, but the medium and larger ones have got smooth muscle in their walls. And I think I talked about the orientation of that smooth muscle. By going back to the autonomic lectures, the smooth muscle cells are arranged around the circumference of the blood vessel. So if that smooth muscle contracts, that will constrict the blood vessel, it will make it narrow. How does that help? Well, if the blood vessel is narrow, less blood is flowing through it, so it's going to reduce blood loss. If you have a garden hose with a hole in it, if you can make it narrow, less water is flowing, and the amount of water pouring out will decrease. So that's the first one. And that's important. It happens fairly quickly after a blood vessel is damaged. But in most cases, it's not enough by itself. If the pressure within the hose or within the blood vessel is high, that can force the vessel open again. So it's good, but it's not enough. And the main ways, the numbers 2 and 3, which I will focus on, are these two here. One, which called primary hemostasis, or platelet plug formation. Obvious role there for platelets. They plug up the hole in the blood vessel wall. But again, we'll see that that is often not enough in itself. This platelet plug, and I'll describe how it's formed and so on, but it's a relatively delicate structure. And again, if the pressure inside the blood vessel is high, it will tend to wash away this delicate plug. And you really, in most situations, need also number 3, secondary hemostasis, which is what we mean, or what we mean by that is when we talk about blood clotting or blood coagulation. This is what we mean, the formation of a much firmer clot, which is stronger and more effective. Okay, so first of all, as I did for red cells and for white cells, just to go back and remind you what platelets are, where they come from, and so on. From the same stem cells in the bone marrow. So here's a stem cell in the bone marrow. And also within the bone marrow, these stem cells will start to undergo differentiation in the presence of the appropriate controlling factors, and they will turn into rather strange cells called megakaryocytes. These are large cells and they've got multiple nuclei. So I presume they're named from the fusion of several cells. And then what happens is that bits of the cytoplasm of these megakaryocytes buds off. And it's these budding off, or budding off bits of cytoplasm that actually form, that are the platelets. And each of these megakaryocytes can produce, I don't know, probably a few thousand, a couple of thousand maybe platelets. So lots of platelets from a single megakaryocyte. And these platelets then go into the blood and circulate in the blood. So what do platelets look like? Well, they are small structures, bits of cytoplasm. They're about 2 micrometers in diameter. So in comparison to a red cell, remember, was coming about cell was a certain size. White blood cells are somewhat bigger. But here's the platelet, about 2 micrometers. Now, it's a little cytoplasm, but like a red blood cell, first of all, it doesn't have a nucleus, because it's coming from cytoplasm. But unlike the red blood cell, platelets do have lots of other things inside. A red blood cell essentially has nothing except hemoglobin inside. Platelets, although they're very small, they don't have a nucleus, but they've got mitochondria, they've got various tubules, they've got various granules, they've got things on their surface, they've got stores of glycogen. They're busy little, I shouldn't strictly call them cells, but what else? Cell-like structures. What do these various granules and so on do? I'll put this one up over here because you don't need to worry too much about this. But if you ever do wonder where something in the whole business of hemostasis is coming from, and if somebody goes and read, okay, platelets produce such and such, you wonder, well, how can they do this? Well, this tells you that some of these granules, the alpha granules within the platelets, produce a whole list of things. These are usually relatively large molecules. Some of them don't mean all that much maybe at the moment, but we'll come to some of these. I'll be talking about some of these. And then the dense granules produce relatively small molecules, ATP, 5-HT, calcium, and so on. Again, a lot of these do play a role in hemostasis. So I don't think you really need to remember exactly what's coming from where, but just to get the idea that these are important, these granules, and also some of these surface phospholipids and surface glycoproteins. These are important in the role of platelets and the role they play in hemostasis. So what role do they play? That's what that's leading to next. And again, a possible way to help you to remember this, previously we had the 3 R's for what B cells and T cells have to do. This is the 3 A's for what platelets do. They stick, they adhere, they become activated, and they clump together, they aggregate. So what we're looking at now, we looked very briefly at vasoconstriction. We're looking now at number 2 on that list, primary hemostasis, the formation of a platelet cloud. So you've got a blood vessel, the wall has been damaged, platelets come along to that hole, plug it up, and that will help prevent blood loss. So adhesion of platelets, they stick to the damaged vessel wall, they become activated, they change their shape, they secrete various substances, these things that are present in the granules and so on, and be released from the platelets, and they play a role in potentiating this whole response. and then the platelets stick to each other and literally plug up the hole. It's very straightforward. The question then is, how is all this brought about? So the next figure, number 54, is a cross-section of a blood vessel. And a sort of small artery or an arterial, let's say this is. So in the center here, this space here, this is where the blood is flowing in or out of the plane of the picture. The lining of all blood vessels is made up of a layer of endothelial cells here, the ones in green. Beneath the endothelial cells, in this open space here, this is called the subendothelium, you have various connective tissue structures, and the ones that are particularly relevant here, in this subendothelium, in this connective tissue, you have collagen molecules, and then surrounding the subendothelium, meaning literally just beneath the endothelium, around that you have one, as shown here, or more layers of smooth muscle cells. So those are the smooth muscle cells that they contract, it'll make the vessel get narrow. So, okay, why am I focusing here on collagen? If this blood vessel is not damaged, let's say this hole wasn't here, a normal blood vessel, platelets, like all blood cells, will just flow through the lumen, through the space in the center of the blood vessel. The lining of the blood vessel normally is sort of Teflon coating. It's non-stick. But when damage occurs, this means that subendothelial collagen fibers become exposed. The blood becomes exposed to the subendothelial collagen. And this is very important because platelets can stick to this subendothelial collagen. You don't want them to stick, and they don't normally, to the endothelial cells, but they will stick to the subendothelial collagen. So what I'm going to look at next in a bit more detail is taking just this area like that, where we're going to look at the relationship between platelets and the collagen in the subendothelium. This is what you're looking at in this enlarged version in figure number 55. So we're looking first of all at how the platelets stick to the wall of the damaged vessel. Here's the collagen, which is exposed. Here are platelets. Platelets apparently can stick directly to collagen, but this adhesion is greatly improved and really it becomes effective, I think, in the presence of a very important protein called von Willebrand's factor, VWF, the red here. This is a very big protein molecule, and it forms a sort of, it's like putting a bit of glue on the surface of the collagen, which greatly increases the binding, because there's various receptors and binding sites and so on. So platelets bind through the von Willebrand's factor to the collagen. And it may be that you've never heard of von Willebrand's factor. It's not something that you hear a lot about, okay? But it is said to be very important, and probably the problems with this factor are one of the most common causes of bleeding problems. So even if you don't hear much about it, it is extremely important. And once the platelets start sticking to the collagen through this dab of glue, they then need to they undergo changes, they undergo some activation, and we're going to come back to what substances are released in a moment, and then they start to build up. So you don't just get one layer of platelets, you get others binding to the first ones, and then others binding to the second generation, and so on. And these are linked together by certain proteins, and the one I've shown here is one of the plasma proteins called fibrinogen. There are receptors for this on the surface of the platelet, on the surface membrane of the platelets, and this then allows you to build up more and more of these platelets, and you form this aggregate, or this plaque. Okay, so I wanted these steps sort of are all happening more or less at the same time. Well, they have to stick first, and then they undergo this, and then this. But I want to just come back to the middle one there, the activation step. It says that they release various substances. What are some of these, and what do they do? So here's a platelet, or platelets that have become activated. They stick to the collagen, to the BWF. They become activated, and they release various molecules. And I'm just highlighting here 4 such molecules. 5-HT, 5-hydroxytryptamine, also known as serotonin, something called thromboxane A2, which, if you're familiar with prostaglandins, is a member of the prostaglandin family. ADP, not ATP, not AMP, but ADP, adenosine diphosphate, and also the phospholipids, which are on the surface of the platelets. These, in a long scheme, give rise to something called thrombin. 5-HT and thromboxane A2 are potent vasoconstrictors. So I told you a few minutes ago that vasoconstriction occurs, but I didn't say how. Well, part of it is just a response to the actual damage. You stretch a blood vessel, you pull on it, damaging it. It's got its own sort of intrinsic response of contracting the muscle as part of it. But this is there's a sort of positive feedback here, because you get more vasoconstriction due to the 5-HT and thromboxane A2. ADP and thromboxane and thrombin cause further platelet aggregation. They increase the stickiness of the platelets, presumably by increasing the number of receptors on the platelet surface that are able to bind to the thrombinograms and the fibrinogen and probably other molecules too. So all of these then are helping to further the vasoconstriction, or help maintain it, and also to form this platelet clump. I'd like to just squeeze in this figure here, which is largely for interest. Have you heard of economy class syndrome? It's a term that apparently should actually be used with caution, but for quite a time now, people have thought that if you travel long air journeys, particularly if you are in economy class, don't have much room to move around, it's more likely that you get clotting in deep veins in your leg. Several hours just sort of sitting there, crunched up, you've got problems. And it's been suggested that if you give somebody takes aspirin before they go on this flight, then this can help prevent this problem. And I want to just very quickly explain why that is, but as not everybody believes. I mean, what's happening on this figure is correct, but whether it really helps you when you're on an airplane is another question. So this thromboxane A2 is a vasoconstrictor, and it increases platelet aggregation. So it's pro-hemostatic, is favoring the formation of blood clots, platelet plugs, and blood clots. But there's another member of the prostaglandin family called prostacyclin, which has exactly the opposite effects. Okay, both of these two, thromboxane and prostacyclin, are derived from membrane phospholipids through pathways we won't worry about. In platelets, there's an enzyme called COX-1, cyclooxygenase in endothelial cells, COX-2. Platelets produce thromboxane. Endothelial cells produce prostacyclin. And this is part of the reason why we don't normally get clotting in undamaged blood vessels. Aspirin blocks both of these, but it's said to have a bigger effect on COX-1 in the platelets. So the idea here is that by giving aspirin, it will prevent the formation of thromboxane A2 more than it prevents the prostacyclin production, and so it will help to prevent clotting under the circumstances that I described. So the theory there is fine. Whether this really does help any more than, being well hydrated, not drinking lots of alcohol, and moving around and so on, is still up for debate. But it's an interesting application. Okay, we'll go on with this next.